So hello, um, my name is Muhammad Ashraf, and I'm from DPM student. And today, I would like to share a little bit about biotechnology in agriculture. And the topic that I choose is tissue culture. So first, we go to the biotechnology. Biotechnology is technology that utilize biological system, living organism or part of this to develop or create different products. Brewing and baking bread are example of process uh, that fall within the concept to biotechnology, just like use the yeast, the living organism to produce the desired product. So next we go to the agricultural biotechnology. The agricultural sorry the the agricultural biotechnology focus on developing genetically modified plant to increase crop yields or introduce characteristic to those plant that provide them with an advantage growing in regions that place some kind of stress factor on the plant, namely weather and pests. In some of cases, the practice involve scientists identifying a characteristic, finding the gene that causes it, and then putting that gene within another plant so that it gains to desirable characteristics, making it more durable or having it produce larger yield that than in a previous. So the biotechnology is important in agriculture sector. Civilization, civilization began with agriculture, and though humanity has changed significantly, agriculture still remain very important. In certain in certain countries, its significance is more obvious, but the reality is that every country in the world depend on agriculture for one thing or other. Here are few reasons why agriculture are important. Firstly, the main source of raw materials. Second, important to international trade. Third, provide employment. Next, help heal the environment. And the last one is the source of our food supply. Next we go to the tissue culture. So tissue culture. Tissue culture, a method of biological research in with fragments of tissue from an animal or plant um, transferred to an artificial environment in which they can continue to survive and function. The cultural tissue may consist of a single cell and population of cell, or a whole, or a part of an organ. So next we see the process of tissue culture, and as sorry, and we can see the first stage. the second stage, and the third stage, also the fourth stage. We have a fourth stage of process of tissue culture. So we go to the stage one, the establishment of aseptic culture. In this, sorry, in this stage, the selected plant are prepared for culturing process. Purpose. The preparation includes cutting the selected source of explain to a suitable size that should fulfill the goal of culturing. The explain are pre-treated and sterilized with certain chemicals before they are inoculated in the growth medium. The chemical used to surface sterilize the short tip axillary bud and meristem are 5% sodium hypochlorite and 0.1% mercuric chloride or 70% alcohol. You can also use ppm for the surface sterilization step. The sterilization the time varies depending on the sorry on the procedure and the explained to be cultured. Sometimes the sterilization process is done in combination with other methods, depending on the level of surface contamination. After surface sterilizing the explant, inoculate them on a grow culture medium contain a component based on species to be grown and method to be performed. The explant are generally inoculated in basic moorage height score MS, medium, or an other suitable media. Supplement with grow regulators like auxin and cytokinin, vitamins, and sucrose. The inoculated explant are incubated at 16H protoperiod at 3000 until 10,000 flux like intensity. So next we go to the stage 2. In this stage, the cultural explant are provided with suitable culture media and grow condition for the multiplication of shot and embryo development. This stage require a considerable amount the time for shoot regeneration from explant. Then the regenerated shot are, are excite into a multiply explant for the multiplication purpose. In some cases, many nodal explant are obtained assessing the single shot as the case where only a single shot is obtained from the apical shot. For the multiplication, trichordine rich media in use to inoculate the nodal explant. In a period of 4 to 5 weeks, 5 until 6 explants are produced from a single shot. The maximum survival of the plant provides suitable media supplement with grow regulator and keep them in a suitable environment. So next, stage 3, rooting of regenerated shot or somatic embryo germination. This stage can be called the rooting stage. In this stage, the growth shot are transferred to fresh medium supplemented with auxin to induce rooting in the regenerated shot. The rooting is introduced by individually separating each shot and the transferring it into the rooting. Sometimes should rooting in directly introduced in the soil having high measure. In some other cases, the first hardening of the plant is done and then they are slowly transferred to soil. For example, in somatic abrogenesis, the explants are allowed to form planted and they are transferred to soil for their growth and acclimatization. The hardening medium should be either perlite, peat, and vermiculite. This medium should hold a considerable measure and be maintained at high humidity. Only the purpose only the properly grown planted with roots should be transferred to the soil. And the last stage is acclimatization of transferring a plant to soil. The plant grown in vitro conditions are very sensitive to disease and pathogen attack. The transfer of plant to the soil should be carefully performed. When the in vitro plants are exposed to the natural environment, they have to undergo several variable of culture condition to protect them from sudden shock. Hardening of the planted are performed avoid stress, measure, and several disease. Also, during the acclimatization, the roots of the plant should be well developed and cuticular layer should be formed which make them suitable for the transfer to open field. In some cases, vitrification, the culture is observed, but in which the plant light look brittle, water shocked, and glossy. It can be due to abnormal functioning of stomata, poorly developed vascular bundle, and an abnormal weight. This condition can lead to the loss of many planted. Next, we go to the advantage of plant tissue culture. A clone, a clonite, a plant are uniform in size and shape and are genetically identical. Tissue culture a process are, are unaffected by seasonal charge. A can plant can be produced all year round and plant derived from a disease indexed mother plant maintained to the same status as the mother plant. Health. You will not have to worry about your new plant having disease since plants produced by the tissue culture process are often free from disease. And variety. New varieties are produced, cultivated, and introduced to the marketing, sorry, to the market higher speed, and more. Next, the advantage, sorry, the disadvantage of plant tissue culture. Tissue culture can require more labor and cost more money. There is a chance to be propagate plants will be less resilient to disease due to the type of environment they are growing. It is imperative that before being cultured, the material in screened filtered to pick up any abnormalities could lead to be new plant being infected. While the process rate in high, if the correct sorry, the correct procedure are followed, success with the tissue culture is not a guarantee. There is still a chance that the process trigger a secondary metabolite chemical reaction, and the new explant or cells grow get stunned or even die off. So next we go to the application of plant tissue culture. The commercial production of plant uses a potting, landscape, and florist subject which use meristem and shot culture to produce large number of identical individuals. To conserve rare of endangered plant species, a plant breeder may use the tissue culture to screen cell rather than a plant for advantageous culture. Large-scale growth of the plant cell in liquid culture is a bioreactor for production of available compound like plant derived secondary metabolites and recombinant protein used as a biomacuticle to cross distantly related species by protoplast fusion and regeneration of the novel hybrid to repeatedly study the molecular basis for psychological, biological, and reproductive mechanism in plant. For example, in vitro selection for stress tolerant plant. to po- sorry to cross a pollinate distantly related species and then tissue culture the resulting embryo which would otherwise normally die. For chromosome doubling and introduction of a polyploid, for example, double sorry double haploid and more. This is usually achieved by application of antimitotic agent such as colchicine of originally sorry as a tissue for transformation, followed by either short-term testing of genetic constructs and regeneration of transgenic plants. Curtin technology, sorry, certain technique such as meristem tip culture can be used to produce plant material from viruses stock such as sugarcane, potatoes, and many species of soft fruit. So this is the example of plant tissue culture. Plant important to developing countries that have been growing tissue culture are um, such as oil palm, planting, pine, banana, date, and jojoba, just like eggplant, pineapple, and more. The the this application is the most commonly applied form of biotechnology in Africa. And the last one, the summary of biotechnology. The biotechnology is a set of techniques used to adapt for specific need of opportunities. Situation that combine multiple needs and opportunities are common. For example, a simple a single crop may be required to provide sustainable food and healthful nutrient protection of the environment and opportunities for a job and income. Finding or developing suitable plant is a typical higher sorry is is typically highly complex challenge. A tissue culture the use involve the use of small pieces of plant tissue which are cultured in a nutrient medium under ster- sterile condition using the appropriate growing condition for each exponent type. So that's all for me. Thanks for watching and thank you. Bye.